So today I was going to talk about establishing a natural history study for AHC, in particular, the importance of natural history studies in rare disease, our experience in doing this in another rare disorder that has features of dystonia and Parkinsonism. So I'll just start with, so the number one interest is in preparing for clinical trials, right? And so I work with basic scientists. There we go. And why is this so hard for a rare disorder? Well, traditionally, if there's going to be a clinical trial, like let's say for a blood pressure medicine or something, there's always a group that gets placebo and a group that gets the medicine. But for disorders that are so rare, it's very difficult to say, oh, half the participants are going to get placebo. And so now, in recognition of this, if you look at what's happened for another rare disorder that affects children, spinal muscular atrophy, very detailed data that identified quantifiable measures was identified in a natural history study prior to the development of any treatment. And so the data was so carefully collected that it was used as the control, so that the children in the study could all get the treatment. So that's really kind of the goal. Okay? And so what do we want? We want measures that are quantifiable, right? And that change over time. We want measures that are both objective, like, so for instance, how strong is someone's tongue in movement? We want patient-reported measurements, traditionally questionnaires, right, that we give to patients and families to fill out, and also clinical objective measurements, which are typically neurologic scales that we use, and that have been validated in related conditions and used over many years, so that we do know that they're useful. Okay. So we can go. Okay, so these are our goals. So the first goal is to identify those quantifiable measures. And the questionnaires that we ask patients and families to do, to fill out, are very important, because the FDA is very focused also on patient-reported outcomes, right? Not just what the physicians see. And then the next question is, so what I've proposed at this point is to collect samples, right? So every time we evaluate people, we ask to collect samples that are easily accessible. Most commonly, what we've used is blood, which is then processed in different ways for different future assays. I also throw urine in there because it's easy to collect, right? And although in our other work, it turns out that we haven't used it, it's just always good to be able to collect it. And these are also the samples that we have permission from our ethics committee at Mass General to collect, specifically in AHC. And again, that is also why I include saliva, right? Because that's easy to collect. You just ask people to spit in a tube. And so then the question becomes, okay, what do we look for? Which is part of yesterday's discussion, right? This is an area that for now I have not specified, because for some of the work that I'm going to talk to you about later, in another rare disorder called X-linked dystonia Parkinsonism, we're going to be doing some preliminary biomarker assays in the next few months. And I'm hoping that combining that with what's also been published by other investigators will help us figure out, and also the scientists who are doing work specifically in AHC will then be able to figure out what's a list to assays that make sense. And also some of the work that we'll be doing in this other, in XDP, is done in-house, meaning, so Mass General has the facilities based on work they've done in Alzheimer's disease to do some of these assays, and some will be sending out to companies. So we will also have some experience about, well, what's a good company and what's not, right? So, and so then I list Parkinson's disease, right? Because there are some features of Parkinsonism that we see in these rare disorders. I listed spinocerebellar ataxia 3 in particular because there are some kids with AHC who have ataxia. This is one of the most common forms of genetic ataxia where large populations, relatively large populations, have been studied. Unfortunately, I've started to dig through the literature, and there's not a whole lot out there about SCA3 and what people have looked at, but there is a National Ataxia Foundation. I have colleagues who do research in this, and so that's also a good resource to go to think about what would make sense to look for. All right. So these are the two goals. Okay, so to explore and establish these, objectively rated measurements, and also have patient-reported outcomes as well. And then for the objectively rated measurements, there are really two parts. There's the part that like the physician does, right? Like we have you do things, we watch how you do it, we give you a rating. But then we also want stuff that we can actually truly measure, right? Like so for instance, having people, we have a tool where we can look at how much pressure you can generate by making a seal with your mouth, how strong your tongue is, because that's like an objective measurement, right? You're not dependent on the neurologist, and having two neurologists, for instance, Dan and me agree that something is mild and not moderate, right? Okay. So who has access to all this stuff? So what I propose is, so there's going to be a searchable database. What has yet to be determined, because I want there to be more searchable fields than there currently are, right? So Dr. Swoboda has kindly given us access to the database and the samples that she left at Mass General, right? So we know where the samples are. We're collecting them. They are housed in a couple different laboratory buildings at Mass General, right? And then my goal is to expand the database because it would have more features in it and make it more easily searchable. Okay. And then both the de-identified data and the de-identified tissue samples are available to investigators. The only caveats to this are, like the investigator has to be at an institution where they are allowed to use these human-derived samples, so it can't be like somebody who's working out of their garage and wants to just like try something with a couple samples. But, so there are regulations in place. Okay. So this is basically saying the same thing. So this is the goal, right? So you're going to get this detailed there will be detailed phenotypic analysis. We're also going to be asking for medical histories, for access to, we sit down and go over your medical history, and then we ask for a release of records so that we can collect all that data. I've also, just yesterday, we were talking about, really diving into the seizures, right? What type of seizures, how do they change over time? That involves getting the notes from the epilepsy doctors and also getting the actual EEGs, right? And so a lot of this is organization and logistics, making sure that we have all the paperwork in place, collecting, downloading the EEG files, all that sort of stuff. And so what's most important, but not at all glamorous, is maintaining this database and expanding it, right? I find that usually is, there needs to be someone dedicated to doing that. I trained in the days of paper charts, so I find it's better to have a younger person who's like more, experienced and knows how to do that kind of stuff. Oh, so this is just telling you the locations, right? So the physical locations of the samples are in a couple different buildings that house research labs, right? Where Lori's lab is, where my group has their offices, and then where Dr. Swoboda was, there's still some, we were just talking at breakfast, there's still some samples that we'll pull, and so that we have everything together. And The database is maintained by the, it's behind the hospital's firewall, so it's protected as much as I think any organization can protect their computer data and stuff, so that it's not, so no one can, only people who are allowed to can have access, because it's in the database that people's names and data are hooked together. This is what I propose for the enrollment criteria. So this is what I put in our protocol that got approved, because my thinking was initially to be as broad as possible, right? And so it's paroxysmal episodes alternating between two sides, or episodes of quadriplegia, and evidence of background abnormal development, or a family member of someone who is affected, right? So that allows us also to collect data and information, in particular from parents, right? And anyone else who is
joined the meeting.
Right? The participants have to be 29 days of age or older. And because we're particularly in the children, plan to offer, the use of EMLA cream, right? If they don't want for a blood draw, just to make it more comfortable. So we have to make sure that no one has an allergy to it. This is just to give you an example of all the data that gets collected, right? So first of all, for every feature, every part of the evaluation, there's a case report form, right? The reason why we're checking off boxes as we go through this is that so that we know we completed everything. If something was not completed, we have to say why, right? And this is all stuff that the FDA requires, right? So you can't leave something blank on a form, right? If we didn't do a particular part of the evaluation, we have to say why, right? Like, the participant refused, or the participant could not stand safely, something like that, right? And then we always draw a pedigree. Anytime we talk to you, I've certainly been guilty of this. Someone will casually say to me over dinner, "Oh, my uncle just went to see a neurologist," and I immediately say, "Why, what for, and will they talk to me?" Right? Because I just want more information. Right? So then we have forms for every follow-up visit. So we also systematically collect the data. I will tell you, yes. And we always update medicines, what's happened since the last time that we saw you. I think a number of you have met, like Trisha, Gabby, and Caroline, who are here. They are like a great team. I'm the person who tends to skip stuff, so they are here to make me do it. Make sure I go through, that we collect all the data. And then the other thing that I threw in here that is still vague, because we have to, I think, figure out the details, is how do we collect all the data regarding seizures, and what's the most useful data that that epileptologists want to see. These are some of the scales. So in collaboration with Dan Sirica and Allison Brashear and Lori Ozelius, who are all part of this ATP1A3 study, we meet on Zoom periodically, and we met in person at the end of April, because I wanted to make sure that we start with collecting data that's applicable to compare across all different ATP1A3 gene mutations, right? So these are the scales that we're starting with now. And I will also tell you honestly that these are the scales that we've started today, because these are questionnaires that I can access without having to pay a fee, right? There are many more detailed cognitive and developmental questionnaires that would be quite useful to do, but they require licensing fees. So I haven't started those yet. So, right. And then the other question becomes, what is best at what age in childhood? And so we've started to talk with cognitive specialists, pediatric psychologists, to get a sense of what makes sense at what age, right? Because there are some very good validated measurements out there. We just need the expertise of people who know the best before we decide what licensing fees we're going to pay, basically. So we're doing this every 6 months. The goal is to do an every 6-month visit. And so that brings up, so my initial thought for this was, okay, so we collect data every 6 months over a 2-year period. Why did I pick a 2-year period? Well, no, in my experience, and I've never worked for a pharmaceutical company, but directly, right? But in working with them on trials and things, I've always been in an academic institution. They're not interested in funding long-term studies, right? They want to do, ideally 12, from what I've seen, 12-month trials or maybe 18-month trials, right? So over a 2-year period, we need to see how people do over a 2-year period. What are the measurements that reliably change over that period of time? Because that's what people are going to, or that are abnormal but consistent over 2 years, because then those are the measurements that can be incorporated in a clinical trial that's 12 or 18 months long, where companies look to see if their treatment helps or not. And so based on previous work in XDP, the rare disorder found in the Philippines, we identified those gentlemen also have speech communication difficulty. And so based on that, we've used these questionnaires that both look at communication and how it's affected on a day-to-day basis, and also food intake. Does someone need to eat soft foods? Is someone completely dependent on a feeding tube to get their nutrition, right? And these are relatively fast questionnaires, 10 questions each. We're also trying not to make this stuff burdensome for people. So this, I don't know how much of this is seen, is just to show. So there's enrollment, and then a 6-month, 12-month, 18-month, and 24-month visit. At every visit, we do the standardized exams, questionnaires, right? At enrollment, 12 and 24 months, we also try to collect some biosamples, right? As much as possible based on people's willingness and tolerance. The reason for that is because we also want to have serial, like blood samples, to see if there are changes in any of these protein markers during that time. Because, ideally, if there's a protein, if there's something abnormal in the blood that is worse as a patient gets worse, then pharmaceutical companies don't have to pay for neurologists to do exams, right? Because that's like way more time consuming. They can just get a blood test to follow if someone responds, right? It's like the neurologic equivalent of checking someone's blood pressure, right? If you want to see if a blood pressure medicine works, there's a really simple, straightforward way to check. You just check people's blood pressure, right? So the goal is to get the simplest, most straightforward way to have measurements that can be used to see if a drug works for a given condition. And sorry, so I just pulled these from the actual protocol that we have that was approved, and we can email this whole PowerPoint slide out to people. And this is just because our ethics committee they want to know what's happening at every visit. And so this is the example of, what do we do? Pre-visit, we try to do like via phone or now via Zoom, right? To chat with people, get as much information as we can before so that the in-person visits are also not so long and cumbersome. Okay, so now I am not a statistician, right? I took one statistics course in college, but I've been very lucky to work with biostatisticians at our School for Public Health, who very much helped us in analyzing the data that we collected for another disorder. And what we did was, for each of the measurements, whether it was a questionnaire, whether it was the scales that I fill out, right? They looked to see from enrollment from day 0 to 18 months, was there a change in that measurement over time? So that was the first thing they did. Okay. And then we identified a series of measurements that did change significantly over 18 months. Okay. So now I'm going to tell you about that work that we did in X-linked dystonia Parkinsonism as our example, right? This is also a rare disorder. It's found in people in a relatively isolated part of the world. And we were able to find data points, measurements that changed over time with a total of 29 men. So now, of course, it depends on, the bigger the change, the fewer people you need in a study. So at this point, like for AHC, we don't know, but this is just to give you an example that you don't hopefully need a whole lot of people. So let me just give you a little background. So this disorder was unique to men whose mothers come from one island of the Philippines. So we know that we knew that it was on the X chromosome. It was first recognized and documented in the 1970s. And these men have features of dystonia, which you can see in that hand drawing, right? Because in the 70s, right, the doctors would go from Manila to this island. There was no video cameras, right? So they were drawing what they saw, right? So these men have dystonia. They have arched trunk, their jaw tends to open, their neck is involved, and they have some features of Parkinson's disease as well. So what did we do? So prior to our getting involved in this study, people knew that there was one area in the X chromosome that was involved, but they didn't know exactly what the gene mutation was, right? And so when I got involved, my job was to collect as many samples as possible. I turned them over to Lori Ozelius and her colleagues so they could, and we carefully detailed who had what symptoms so that they could narrow down the area to figure out what the genetic problem was. Yeah. And this very busy slide is just to show you that they figured it out, right? This is the problem right here. And it's just 6 bases. That whole area in red gets inserted into the gene, but those 6 bases there, the more copies someone has, the younger they are when they get the disease, right? Is there anything you want to add, Lori? No. Okay. So what's interesting about that is, right, so there it is. We know what the genetic problem is, and it's something that theoretically could be cut out of the gene. So while my colleagues were doing very sophisticated work in the lab to try to figure out if in fact we could safely cut that part of the DNA out, my job was to figure out, what are the clinical measurements, identify patients, organize them, figure out what the clinical measurements are that could then be used in a future trial. And so that's why we started this natural history study in that disorder. We use some of the same scales that I've talked about today. And we look to say, is there a change from enrollment to 18 months? So if you take all those scales, right, like each scale has different parts. So we actually had 99 different things we were asking people or evaluating or measuring. This is just to show you, right? So each part of these scales has different questions. So the Parkinson's rating scale has 2 parts that the families and patients answer, 1 part that the neurologist does. The dystonia scale has 1 part that the neurologist does, a 2nd part that the family and the patient does. And then there are things that we can actually get hard numbers for: lip strength, tongue strength. How quickly can someone say certain syllables within the span of 10 seconds? Okay? Because those are hard numbers. You can measure that stuff. We then gave all this stuff to the statisticians, and they did their analysis, and they found that out of those 99 measurements, there was a combination of 9 measurements where there was a significant change over 18 months. And what's really interesting is, right, so what are the measurements? Weight. So in this particular disorder, men have a lot of trouble eating, chewing, and swallowing, so they do tend to lose quite a bit of weight. But look at the others. Most of these are questions about disability, things that the patients report. right? Their ability to eat, their ability to get dressed, their ability to shower, bathe themselves, right? There's very trouble with eating, food sticks in throat. They experience that their speech is a problem, so it's hard for other people to communicate with them, which is incredibly socially isolating, right? When that happens. There are very few things here that require a neurologist, which is both good and bad for me, right? Like, right? Which is, if you look, it's facial expression. That's something that the neurologist rates. Freezing of gait, and the quality of speech. Anything with a 3 in front of it are the parts of this that require the neurologist to do a rating. And so the interest now is, okay, this is great. We could cut down our evaluation because it's a lot to ask of someone to go through all the questionnaires. Like, could we just now only look at these things over 18 months, right? Well, first, what we're currently in the process of doing is we want to see if we can reproduce this, right? So we're doing all the same 99 measurements in a whole new cohort in the Philippines. That we should finish collecting that data by like March of next year. It'll then be analyzed by the statisticians, and hopefully we will see that some of the same measurements show up, and then we'll have a really strong argument. Like, look, we don't have to do all of those 99 questions and measurements. We can cut it down to a minimum of 9. I'd be a little nervous to do just 9, but maybe we'll get it down to like 20 from the 99. So let me see if I can explain this properly. So one of the other questions that we asked was, okay, we can get it down to 9 measurements, but does that really capture the full spectrum of the symptoms, the physical problems that people with XDP in this case have, right? Or do we really need to broaden it a little, right? Like, how's the best way we can capture the full range of communication and eating problems, the full range of physical problems? So again, we asked the statisticians to look at this data in a different way, like what are the measurements that overlap? So what they did was, so wherever you see this kind of orange color, where it gets, where you see these squares, sorry, I'm short and can't, but like here and here, these are where measurements kind of overlap together, right? And so then they said, okay, let's pick measurements from each of those squares and say how much, if we combine them, how much of the total change over 18 months do those measurements tell us? And then they said, okay, if we take 21 of the measurements instead of just so the 9 is like the bare bones, but if we take 21 of the measurements, that explains about 60% of the variance, the change over time that we see in 18 months. And this is the best that I can explain it to you. We should definitely get Jeff Miller, our statistician, to correct any errors that I have made in the last couple of minutes. But so then the argument becomes, we should always get these 21 different measurements, right, in a subsequent study. And so that's this kind of proposed minimal battery here. It has the same 9 measurements, and it also has some more. So that is what we're doing for that disorder now. That is, our experience there, the experience of our colleagues who've done remarkable work in spinal muscular atrophy is really what led us to do this, right? So that's kind of the roadmap that we're following. And the other point I want to make about the biomarkers is, so we've come up with a list based on what other people have reported for disorders that have similar features, and also based on the work from basic scientists who are specifically working on this XDP mutation. So then I would say, based on the cellular data, the basic science data that comes out of the AHC mice and cell lines, that should also inform the list of biomarkers. And so based on that, that's why we're asking, for anyone who's willing to participate in this study. And we're more than happy to coordinate with other organizations. All we have to do, and we can share de-identified data and samples. There is always paperwork involved. It's, to share that stuff, but that way nobody gets in trouble. Everybody fills out the paperwork and we'll share all that de-identified data and samples. And that's our goal. So that's all I had. If you guys have, I'm sure there are questions, comments. Thanks.
Okay, this is a logistical question. So as you know, medical records are extremely difficult to come by. My girl was in 3 different hospitals in a couple different states. One of the things that we learned, she's had several EEGs, continuous EEGs, but every hospital uses a different system to read EEGs, and doctors don't always just accept the EEG notes. And so they always want to do their own EEGs, which for AHC kids, for my girl, that's stressful and that doesn't help the situation. So if someone's willing to do this, right, how do you get all of the EEG studies? It takes about 10 to 20 phone calls just to get neurology notes to another neurologist. So I'm just thinking logistically, how does it help you guys? What do you actually need? Do you need the videos of EEGs? Do just the EEG notes help? And then on top, I know, long question, sorry. On top of that, right? So for instance, my girl, they said she was having a generalized seizure on the EEG, but neurologist reviewed it after I said, no, it wasn't a seizure, and it wasn't actually a seizure. So you have to have the neurology notes about that EEG on top of the technician's notes. Anyway, so there's a question somewhere in there.
Yeah. So this is, yep. So this is the whole, so it's all about like organization and logistics. Like I can't even tell you. So the way that we do this, and we were having a conversation last night about getting EEGs. So we're very used to this. We get you to sign a medical release form. We send it to all the different places. We then have to call them multiple times to bug them, right? Because they're like, records places are overworked. They probably just have one person, working in the department. Some hospitals have asked to see a copy of our ethics approval letter as well. So we send that. But it really, what it requires is a person who's dedicated to just like making calls and following up and, like having a list of all the stuff that's missing, right? Which as soon as we have your signature on the forms, we can do. Now, one thing I've learned though, is some hospitals, a number of them now, they'll only honor a records release form if it's been signed and dated within the last 12 months. So sometimes we come back to you guys, we're like, hey, and you're like, why do you want me to sign this again? Because the hospitals are making us, right? So we start with, and then but we collect all the notes that way, right? So Marcelo and did I pronounce that correctly? We're just talking yesterday about how epileptologists always want to see the actual EEG. So the part that we need to figure out is, there are secure ways to download data, right? Because we do that for genetic data all the time from one institution to another. So what I don't know, but it has to be solvable, is how we download all those EEGs. So then what we would do is we would get that downloaded securely from one institution server to our server, right? You have a number, your patient 456, right? We get it, we download the data at Mass General, we remove your name, we put patient 456, and then we securely transfer it to Marcelo, who actually knows how to read EEGs, whereas I don't, have a wet, yeah. And then the trick with wet signatures I found, because like Medicare requires that for a lot of things, is blue ink. Yeah, absolutely. Yeah. It's like so annoying. Yeah. Right.
Thank you. That was very helpful. I have 2 questions. One, very simply, in your enrollment criteria, I didn't see genetics on there. Will this be exclusively for AHC caused by ATP1A3 pathogenic variants? It's AHC independent of genetics. Okay, I see that. And secondly, I think a natural history study in this disease, I would think uniquely has to capture the paroxysmal nature of symptoms rather than static nature of symptoms. And so I would like to hear how that is being approached. I don't think there's a survey in the world that does that well. And so would be interested in your approach.
So for the inclusion criteria, my thought originally was, let's start by being as broad as possible. And then if we have to narrow it down or group people, we will. I'm also doing that from the strategy of, I don't want to keep going back to my ethics board and fill out other forms for, it's just the fewer forms I have to fill out, the happier I am, kind of thing. So that was the first. And so for this issue of the paroxysmal nature of it, yeah. So here's what I'm thinking about so far, and I think we need to talk some more about it, is first, like, do we have to make sure, well, there are two ways to do this, right? Like, we started by asking families, like, well, when is the best part of your child's day, right? Like, so do we try, like, for kids where it's fairly reliable before they get too tired, there's a good part of the day. Do we start by making sure that we do exams then? Right? And see, so do we have to do the best part of the day over time? I mean, I think we have to do that. Then the question is, what are the triggers? Are the triggers clear? Right? I don't want to, we can't. And then I would say, do we start by, if there are, honestly, if there are videos taken after a trigger that induces an episode, we first start looking at those videos. Right? Because it's not ethical to induce it. That's the problem. So I think that we have to go by asking families to start, which I suspect a number of parents have already done, but to do that and be willing to share those with us. And then, of course, the issue is going to be, it gives us some data. It won't be the same exact video that we do when we do our research visit, but if we can start with that and then figure out what else has to be done.
Yeah, to add to that, I guess we did the survey that you had set up, I think towards the end of 2021. It was a daily survey for 2 months. I think that it was 4 questions and quite simple. So I don't know if that would be applicable here. It wasn't onerous. We got a reminder, we did it. So that might handle the proximal episodes to some extent.
Yeah, I mean, we've spent so much time over the years and this gets so complex, but we do need absolutely for clinical trials to figure out how to agree what to call what and what the duration is and the burden of it. How to do that has been one of the hardest things. Stephen Henderson has an app and we're going to work with Tartamed to sort of see if we could try this out in their study. But I think we do need some kind of a way to sort of do that if we're going to actually look at the impact of any therapy on the frequency or duration of the paroxysmal. I mean, one of the reasons, I was so excited that Nutan's involved. Nutan Dr. Sharma is a movement disorder specialist. There's a lot of different ways and apps to do this, but none of them have really been used very effectively for drug trials, to my knowledge, for paroxysmal, except in epileptic disorders. And so this really is tricky. It's new ground, and we're going to continue to work on this.
Yeah, I guess the question which I've been asking a few of you the last couple of days is like, coming from Canada, is there protocols that need to be in place for these every year or so, giving biospecimens and doing like physical exams and that?
All right, so the way it stands right now is, so we've said in our protocols that we're doing these assessments in people in North America. We go to people's houses, do blood draws there, nothing more invasive than that. So far, nobody's asked us anything more. So I'm assuming that it's okay. Yeah. So, I mean, I've gone to Canada for research study visits for other things. And we've been able to ship blood back to the US, right? Because you can't take anything on the plane anymore. I remember the days when we could, but I don't want to get arrested, but we can we can go onto FedEx and send stuff back. I've also had other people ship to us from South Asia, South America. Now the issue is like everything, you can only reliably get DNA from those blood samples because, customs can sit on it for however long they want to sit on it. But at least the last time we did this, it was like, you just check off a box that says this is for research purposes. It's not being used for clinical purposes. And then it seems like it's much easier to get through customs. At least so far, that's been my experience.
I thank you. If there are any further questions, Dr. Sharma is going to be around, so you guys catch her on the break.